You know. All right. So we left off here on Wednesday talking about that, basically adding another term to how DNA is replicated. Oh, we didn't go over the quiz. We'll do that. We'll do that at the end. Talking about how DNA is replicated. On Monday, we talked about DNA replication being semi-conservative. That after your replication event, each molecule has one old and one new strand. Another way in which the DNA is replicated is what's called semi-discontinuous. So you have one strand at each fork that's replicated continuously because DNA polymerase is allowed to replicate in the same direction helicase is moving. But on the other strand of the replication fork, because of the anti-parallel nature of DNA, the DNA polymerase is not allowed to polymerize in the same direction helicase is moving and has to actually polymerize in the opposite direction. And so it has to wait until helicase opens up enough that primase can get back in there, write a new primer, and then DNA polymerase can come in, pick up where primase left off, write, cut out this primer, and then ligase can come in and fix that nick. But you get these short fragments, and these are called Okazaki fragments, named after the person who discovered this process of semi-discontinuous replication. Okay? Now keep in mind, in any replication event, you get what's called a replication bubble with two forks, one on either side of the bubble. And each fork has both a leading strand, where you have continuous replication, and a lagging strand, where you have discontinuous replication. Therefore, we refer to replication as being semi-discontinuous. Both continuous and discontinuous, semi-discontinuous. Right? Okay, just in the same way that semi-conservative is part old, part new, right? Does this make sense? Okay, cool. Now, it seems to be that all of our different forms of DNA polymerase can cut out nucleotides that are in front of them. Okay? Or, I'm sorry, that's not true. I just told you something that was a complete lie. It was an accident. It seems that all DNA polymerases can cut out nucleotides that are behind them and to correct their mistakes. But not all DNA polymerases can cut out nucleotides in front of them, which is why you get DNA polymerase 1 involved on the lagging strand, but not as much on the leading strand. Because DNA polymerase 1 can cut out nucleotides in front of it, and it can come in and cut out these RNA bases. DNA polymerase 3 is the main polymerase, and it can't do that. Yeah, Dylan.
How many nucleotides does polymerase cover?
Polymerase? Any polymerase? How many? Like just the machine? How big is it? Functionally, it's only covering one at a time, right? Because it's reading one and adding a new one. But in essence, it's probably covering between 5 and 10. Based on its size. But functionally, it's only interacting with one base at any given time. Yeah. But it is a big machine, so it's probably covering between 5 and 10, keeping other machines from having access to it, which is, I think, what your question is, right?
So it can cut out the ones that are, that it's covering?
Oh, no. If it's going to cut one out, it has to do it one at a time by moving backwards. Or if it's DNA polymerase 1 and it's cutting out what's in front of it, it still does it one at a time. Yeah, so DNA polymerase functions on one base at a time, even though it's covering more base than that. That's a good question. Yeah, it cuts out one at a time, either forward in the case of DNA polymerase 1, or backwards in the case of DNA polymerase 1 and 3. And also 2, which we don't know all that much about 2 because it doesn't seem to work as often or as hard as 3 and 1. Yeah.
If that's the origin of replication, then why is it going towards it? So it has two different origins of replication?
No, why is polymerization going towards the origin?
Yeah.
It has to on the lagging strand because that's the only direction polymerase can write.
So that's not the origin of replication for the lagging strand,
Yeah, you're right. Yeah, the origin of replication is here on the lagging strand. But the origin of replication is called the origin of replication because that's where the DNA was first opened up by DNA a protein that opens that up. Or in the case of eukaryotes, is opened up by DNA polymerase alpha, That's more than what you need to know.
So it starts and then it stops when it reaches where the leading strand.
Well, yeah, I mean it'll stop wherever there's no more. So DNA polymerase 1 would come in, would start here, and would write until it's cut out these RNA bases and replace them with DNA, and then it would stop. And it would leave a little nick that would need to come back. Yeah, and then it would go back and attach here and start over again. Yes.
That's why, okay.
Yeah, and that's why it's semi-discontinuous, because it's like right, stop. Right, stop. You know, it's like Morse code, you know, where you have to get that extra pause in there to let them know you ended a sentence. I don't know if you all know about Morse code, but I learned about it when I was a kid because it was awesome, because other kids didn't invest the time necessary to do that, and so nobody knew what my brother and I were talking about, including our parents, until my little brother caved under pressure, like little brothers always do. Ruined our game and my life. All right. So the remaining important questions. Well, that's on the recording now. Hopefully my brother doesn't watch this. All right. What happens if DNA polymerase makes a mistake? Excellent question. What if the mistake isn't fixed? Also an excellent question. You're welcome. How is DNA packaged within the cell? And then how does prokaryotic packaging differ from eukaryotic packaging? And so right now I'll talk a little bit about the quiz. First question, what was the first question on the quiz? Oh yeah, what activity helps in DNA repair? Probably the best way to answer that would just be to say exonuclease or endonuclease activity. But you could just talk about mismatch repair, DNA repair. DNA polymerase fixes most of the mistakes it makes. Some of them persist, and you have some other machinery that are designed to look for mistakes. The second one, it was false. Persistent errors do not lead immediately to the death of the individual, unless those persistent errors happen in very critical places. But I mean, that's the whole idea of where mutations help to lead to variation, right? You get evolution by mutations and natural selection. Those mutations are persistent errors. Okay. And then the three main ways protein interacts with DNA is in copying or replication, in expression, and in organization. But there are a ton of ways you could have answered those questions. Those are the big three that this chapter talks about, and then next chapter talks a lot about expression, but this chapter talked a little bit about it. Those are the big ones, but if you just gave specific proteins like you did on Wednesday's quiz, that would be fine as well. All right, so what happens if DNA polymerase makes a mistake? And I'd like to tell you everything, it just doesn't make mistakes, but it does. And then I'd also like to tell you, I don't know why I'd like to tell you this, that everything just really just breaks. I mean, everything just goes to crap, you know. But that's not true either. I don't know why I want to tell you that, but it's not true. So, yeah, while DNA polymerase can only attach a nucleotide where you have a free carbon 3, many of them can cut out nucleotides in either direction. Not all DNA polymerases, but many of them can cut them out in both directions. All of them can cut them out backwards, can back up and replace the one it put in correctly. And so DNA proofreading, most of this is done, again, by DNA polymerase itself, checks and corrects most of the mismatches. So if DNA polymerase adds a wrong base, DNA polymerase will correct the overwhelming majority of those mistakes. Yeah, Dylan.
If polymerase doesn't correct it, will ligase come in after and correct it?
Ligase won't correct it, but there's what's called mismatch repair, and mismatch repair mechanisms will come in afterwards and fix it. So ligase just attaches, a carbon 5 of one to the carbon 3 with another with a phosphate group that aren't attached. And so there are other machines that will take care of that. And so the 3 to 5 prime, notice this is the opposite of how they write. How do they write? 5 to 3. So 3 to 5 is moving backwards. Okay? And so this is a third function of DNA polymerase. And, in addition to reading and writing, and this is what, again, replaces most of the mismatches. Yeah. And then here's the answer to your question, Dylan. Most of these persistent mismatches that DNA polymerase didn't fix are actually corrected by mechanisms that go in, literally cut out the wrong base, and then DNA polymerase will come back in and fix its mistake. Okay. So DNA polymerase still has to work even if it didn't catch its own mistake. It's got to come back and fix its mistake at some point. And so well over 99% of the mistakes DNA polymerase makes, it catches and fixes itself. Of those it doesn't, 99% of them are fixed by repair mechanisms, by cutting out the bad base and forcing polymerase to come back in and add a new one. And so you're dealing with, probably 1 in 100,000 mistakes DNA polymerase makes actually persisting. Yeah, actually persisting. Yeah.
Why do they make mistakes in the first place?
Because they're writing 1,000 bases a second.
Okay.
And so it's just by the sheer volume of replication, sometimes it doesn't catch the mistakes. And then our genome is 4 billion bases long. And so as machinery are scanning for mistakes, some of them just don't get caught, just because of the sheer size of the genome and the sheer speed at which it's being replicated. That's a good question. Everything's a balance, right? If you're going to emphasize strength, you sacrifice mobility. If you're going to emphasize speed, you sacrifice fidelity. If you're going to emphasize fidelity, you sacrifice speed. But DNA polymerase meets both as well as possible. Okay, so here's the proofreading. Again, remember the sliding clamp and the sliding clamp keeping DNA polymerase physically attached to the strand. And so if it adds in the wrong base, which it did here, it added in the wrong pyrimidine, it added in cytosine instead of thymine. When it tries to move forward, it recognizes, because it's physically attached, that there's a road bump. It's not able to move as forward as quickly or as smoothly as it's used to. So it realizes it made a mistake, it backs up one base, cuts it out, and then puts in the correct base, which would be thymine, the correct pyrimidine. And so that fixes 99% of the copying errors that DNA polymerase makes. Of the 1%, over 99%, we'll just say 99% for simplicity. So of the 1% that don't get fixed, 99% of those are caught by mismatch repair machinery that'll go in and then still recognize that these bases aren't hydrogen bonding together. And it'll, again, do this by kind of scanning it, realizing there's an issue. It will cut out the incorrect base and some around it. DNA polymerase will come back in, attach new nucleotides, leaving a nick here, right? Because DNA polymerase just attaches a nucleotide at a free carbon 3. It gets down here, there's this free carbon 3, but there's already a nucleotide there, so it can't do anything. Ligase comes in and fixes that. Okay, so that's where ligase would come in and will function in mismatch repair. Okay? We're going to take a lecture break. And what I want you to do, working with your group, I'd like for you to answer this question. If mutations are so essential for generating variation, which is the whole reason why we make an argument of why sexual reproduction exists, right? So that you get variation in your offspring, so you don't make just clones of yourself. Okay? So if mutations are so essential for developing variation, why does the cell work so hard to try to minimize the number of mutations? Okay, think about that. Okay, answer that question. Spend a couple of minutes talking about that. And by the way, this is more of a philosophical question than it is a mechanical question. Okay, I mean, you can address it mechanically and be like, well, the reason why it invests so much in doing that is because it can't help it, right? It recognizes that you don't form hydrogen bonds. That's fine if you want to answer it that way, but that's boring. That's boring. And so when your group representative shares that thought with the class, we are all going to be bored. So just putting that out there. Okay? No big deal. If mutations are so essential for generating variation, which variation it's kind of trumpeted as like, not only is this the source of what natural selection gets to work on, but it's usually the explanation for why do organisms behave in this way, right? Why does sexual reproduction exist? Why do organisms, tend to not mate with close relatives? Why do organisms tend to, disperse from their natal homing ground, right? And all of these, we answer it because it increases variation. And variation is wonderful because then natural selection has something to work with. But on the mechanical level, on the molecular level, the cell invests an enormous amount of energy into ensuring that those variations don't come about. Now they do, right? We'll talk about this in the next question. What if the mistake isn't fixed? So this variation does come in, but why does the cell invest so much in preventing something that we use as the answer to why it does so many other things? Does that question make sense?
Yeah.
Okay, 2 minutes, starting now.
So that means that the odds of variation should be small enough. It was the minimum. So it's better to stay the same as forward. Also, there's other ways to do variation. Like how from evolution, variation is caused by abnormalities along the way. God created things zero, which we can kind of assume that they're really. For instance, like there's some things so they're like more genetically versus just so it's probably created that's really diverse and they're close to
Yeah, I think.
so they weren't necessarily the differences were caused by
Yeah, I think that's the question is kind of your question. I think the answer to the question is that mutation is there. Like I think there's a reason that cell tries to genetically actually, like Dr. Newman was saying in our origins class,
All right, you got about 15, 20 seconds to wrap up your thoughts and get a representative.
Yeah, so I think the question, the answer to the question is, yeah, so I think it's a false assumption to start.
All right, y'all have a representative? You know there were 7 people missing on our last group quiz Friday. Now granted, those 7 people didn't know it was going to be a group quiz Friday, but there are only 4 people missing today. Still 4 people.
How many people in the class?
How many? 32. How many people were in this class when the semester started? It's a different question altogether. The answer to that is 36. All right, so we're going to start here and we're going to move counterclockwise. So group 6, what are your thoughts?
So our thoughts were that mutation is kind of a trick question because mutations aren't actually essential to diversity since they're the number of helpful mutations are like really, really small.
Mm-hmm.
What we studied.
Sure.
And so the odds of it being beneficial are really small.
Uh-huh.
And so once to minimize them, because it's better to stay the same than it is to like possibly get better, but more likely get worse. And there are other mechanisms for diversity,
Such as?
Sexual reproduction. So like things being placed really far apart on the genome, so
Okay.
they can switch easily. And
Yes, so what you would call those are epistatic effects.
Yes.
When the product of one gene influences the product of another.
Yeah.
And so it doesn't necessarily need to be a new, a new genetic code. It could just be a new combination,
Exactly.
right? Yeah. Like, let me give you an example. I was born with a widow's peak, but I was also born with a genetic predisposition for male pattern baldness. And so my widow's peak disappeared, and that would be an epistatic effect, right? You're like, okay, well, I didn't generate anything new, but I erased something that was there in the first place. So I've got, compared to my family, my brother seems to have lucked out with the whole male pattern baldness thing. So he's still got his widow's peak. So based on us, you're like, wow, that's some cool variation. He's got a widow's peak, he doesn't. No, we both do. My hair's gone, so you don't see it anymore. Yeah. Group 5. You didn't get a you gotta do that before.
No, it's fine. I can do it.
We were still talking about it.
Okay, I respect that.
So kind of piggybacking off of what Alyssa said, obviously there's certain mutations that are not favorable in the majority.
Sure.
Are not.
Yeah.
So, kind of going off this idea that, structure affects function, and even small changes can drastically change function.
Mm-hmm.
So, there's reasons that mutations are obviously helpful because we as humans need to adapt, but that's probably the one reason that I would say.
Yeah, I think what you would find is you will be hard pressed to find a mutation that is undisputably beneficial, right? I mean, there are some that are proposed, and there are some that you can make a good argument for. But yeah, I think you'd be hard pressed to find that. So it makes sense, right? Individuals that replicate with a high level of fidelity are probably going to produce more and stronger offspring than organisms that replicate with a small amount of fidelity, right? With a lot of mistakes.
Yeah.
Yeah.
Well, even you see with similar genetic lines. So basically, let's say you have a brother and a sister, right, and you have them, breed. You'll see a lot more of genetic disorders.
Yeah, because of inbreeding just makes recessive conditions more likely. Yeah, more likely to actually be expressed. All right, group 4. What do we got?
There has to be like some sort of control going on,
Okay.
or parameters of how much mutation can happen, because if it just starts going out of control, then you're gonna end up with stuff like cancer or genetic disorders.
Sure. Yeah, pretty quickly. Charles, did you have something you wanted to add?
Yeah, as far as what makes variations and change within organisms, it doesn't necessarily have to be mutations like the last group said. For example, you have the wolf versus a chihuahua. That's a huge difference. And it wasn't exactly through sexual reproduction, but we bred wolves all the way down to a chihuahua.
Yeah, it's called heterozygote fractionation. So you just eliminate the variation. That's done with basically the exact opposite. You're not increasing variation, you're deleting it. And as you start to delete that variation, you start to notice some strange things. And then if you like those strange things, like for some reason you want a dog, if you want to call it that can fit in your purse, you're like, I mean, you could do that, right? As you start to eliminate that variation. So that's that with dog breeding, and then also with dove breeding. Dove breeding isn't super popular in the U.S., but it's super popular in Europe. They actually have competitions where people will bring their doves, and yeah, it's interesting. But it's done the same way. It's called heterozygote fractionation, and you are eliminating variation to accomplish that change.
When you say that, also sometimes there's genetic potential that's not realized in the organism, and as
Maybe you would, yeah, possibly. I mean, again, if you want to talk about the Chihuahuas being genetic potential, right?
well, what I mean is like in
I
the,
know. I'm just giving you a hard time.
Okay.
I hate Chihuahuas.
I hate
So
wolves.
I've got a very cold place in my heart that Chihuahuas reside in. I mean, it's really cold, like absolute zero.
Snake, like a boa constrictor, I will feed it chihuahuas.
Oh man, that would be a lot of fun. All right, group 3. Well, I mean, they're way less expensive than rabbits. And so if you have a big snake, you have to feed it rabbits because rats aren't big enough. Chihuahuas are, I mean, you can get them for free. Like, you go on Craigslist and people are, like, begging people to take these Chihuahuas away from them. All right, group 3.
Well, we focus on mutations are just kind of like understood generally. Emphasis on the word generally, not all, most are seen as bad. Most of them are not good. Mutations do occur at random, whereas natural selection is not.
Correct. Yep.
Random. And so, and there are mutations that can occur that can make an organism better fit for their environment, just as there are also mutations that occur that can make it not so fit. And
Sure.
if it's not so fit, then it obviously won't survive, and if it is, then it will obviously outlive and be able to pass on those genetics to its offspring. Another thing that Dylan brought up was mutations just being seen as like as part of the fall, and just seeing it like that as mutations just being a kind of mistake in genetics.
Sure.
And from a biblical worldview, from a creationist perspective, rather, that could be another potential argument. But from an evolutionary perspective, it would be mostly, it just kind of.
Yeah. Yeah, it doesn't seem like mutations are a great tool by which you hang your mechanism of the variation in life on.
Yeah, well, the thing is with genetic variation, that just goes into how large the ecosystem is, in addition to how large the population is. Because if it's a very small population, then obviously there's not going to be a whole lot of genetic variation. Because if it's very, very large, then the statistics are significantly higher for that variation.
Yeah, there is a correlation there, but you got to be careful there because there are species like a cheetah where there are a lot of them. I mean, they're not critically endangered, but if you took 2 cheetahs, there's a pretty high probability they'd be more closely related to each other than you are to your siblings. And yet they're just 2 random cheetahs. So it's a huge number of individuals, but very, very little genetic diversity. And then you have some populations that don't have very many individuals, but there's a lot of variation. But yes, there is a correlation there, and we'll actually talk about that in our mechanisms of evolution discussion. All right, group 2.
You kind of said something that
Okay.
We may not understand fully what causes variation.
Sure.
Just because mutations seem to be responsible for it, there may be something else going on.
Yeah.
And so what the cells are working hard to do is not eliminate variation.
Sure.
Mutations.
Yep. Yeah. Group 1.
We said that God originally created things to be perfect.
Okay.
So that after the fall, mutations came into the world, and mutations generally lead to issues, such as birth defects and cancer and all that.
Yeah.
And so, so they're just trying to continue their function of making things.
Yeah.
perfect.
Yeah, so the overwhelming majority of mutations are what are called nearly neutral. There's no mutation that is probably entirely neutral, but most of them are what we call nearly neutral, which means that their impact on the fitness of the individual is so small that you can't measure it. Okay? Now, there are some mutations that are not nearly neutral, that are actually pretty far away from neutral, and those are very rarely fixed in a population. Those mutations usually are to the detriment of the population, even if they just show up in a single individual and are very quickly weeded out. It's very unusual for a not nearly neutral mutation to become fixed. That is, every individual in the population has it, or at least a good percentage. It would be the norm for that population. Most of those are just completely weeded out. And even with the nearly neutral, most of them are completely weeded out. It's hard to actually fix a mutation, even if it's a mutation that increases fitness. It's very, very difficult to fix that, to make it to where that's the norm for the population. And so all of this is just interesting. Yeah, it makes sense why you have to have this mechanism to protect against mutations, right? That makes sense. But it just presents an interesting question as to whether you can even rely on mutations to produce the variation you need to get the diversity of life from mutations and natural selection. And many individuals are coming to the conclusion that we need additional mechanisms, that mutation and natural selection alone is not enough to explain the variation we have in life. And either that variation was created, which is one view of origins, or there's some other mechanism to generate that variation. Yeah.
Is there like, so I heard about how like built into the DNA are like enzymes that kind of process of like taking DNA from one segment and like moving it to another section in the genome?
You just completely ignore that mutation. Is that what you're getting at?
No, it's not like.
You're talking about just moving material through the genome.
Like, yeah.
Yeah. Do you call those transposable elements?
Is that what they're called?
Yeah, and it seems that most genomes, if not all genomes, have those kind of mobile elements where they move around. They're fluid. They move around within the genome.
And then do those make like significant variation? Like
Potentially, yeah. I mean, they can result. Yeah, you see that in Indian corn. Indian corn, where you get the blue and the orange and the yellow and the white kernels. A lot of the variation in kernel color, which each kernel is like a different individual. It's not like a different individual. Each kernel is a different individual. A lot of that variation is because of mobile elements and not necessarily differences in the genome.
Okay.
Same thing with a calico cat. The different, where you have orange patches and brown patches and black patches and white patches. A lot of that variation is with the mobile elements and figuring out where they're in the genome, and then also which X chromosome gets shut down. So that's another thing we'll have to talk about when we talk about inheritance. And so you've got lots of things going on.
Okay.
Yeah, and apparent variation is not always variation. Is basically what I'm getting at. So you can have two individuals that are genetically identical and yet they're morphologically distinct. And so that variation, yeah, apparent variation doesn't actually mean genetic variation. So, all right. So what if the mistake isn't fixed? We already talked about this. This is a mutation. And these point mutations, these are where you make a mistake in copying a single base. The wrong base is added. It's called a point mutation. And this does fuel some of the variation in living organisms. Is any portion? Now, this statement is incomplete. This should say, if the point mutation is any portion of the DNA that codes for protein, the mutation can be devastating. I don't know what happened to the rest of the statement. I'm going to claim Canvas stole it. And I would like for you to prove me wrong. Okay? No. So if the point mutation is in a portion of the DNA that codes for protein, the mutation can be devastating. Okay? But if it's not in a portion of the DNA that codes for protein or codes for some other active element, then it would most likely be nearly neutral. Not entirely neutral, but nearly neutral. And then here's the question that we kind of hinted at, but we didn't actually, not all groups hit entirely. So what are some other sources of variation other than mutation? Sexual selection, right, can attach some variation because it can accentuate certain features, right, and make it to where you get some features that weren't present before, or at least extremes that weren't present before. Yeah, we already talked about here. Alyssa mentioned where one gene influences the expression of another. It's called epistasis or epistatic effects. And so just, as a picture, a representation of variation, I want to show you one of my favorite forms of variation, and this is due to a mutation, albinism. And so here you have an albino raccoon. Beautiful, right? Raccoons are deceptive because they are really cute animals, but they are probably the dirtiest, most vicious animals that most people ever interact with in their entire lives. They are feisty and they're disgusting. So, but they are cute. They are cute. Here's an example of another type of variation, but is also due to a mutation. So this is a melanistic jaguar. This is a jaguar, but it produces so much melanin, you actually can't see its spots unless you get really, really close, and then you can see its spots. But it's not advisable to get really, really close. And so you have what we just collectively refer to as panthers. But you could have melanistic leopards and melanistic jaguars. So you have panthers in both South America and in Africa. Gosh, I keep kicking these things.
Wait,
Yeah,
so
listen.
some panthers are more related to jaguars than they are to other panthers?
Well, panthers. So then there's the term panther, which mountain lions. One of the names for a mountain lion is a panther, and that's a completely separate thing altogether. But yeah, panthers are melanistic jaguars or melanistic leopards. Yeah, it's not like a separate species. Now, the Florida panther is a mountain lion, and that's a separate subspecies of mountain. It is confusing, but lots of things in Florida are confusing.
So why is it only jaguars and panthers that look similar to each other that are melanistic and not like tigers or cheetahs?
Probably has to do with the way in which the pigment is laid down to produce the spotted pattern that they have. Because cheetahs are spotted as well, but the spotting pattern is different than in leopards and jaguars. Leopards and jaguars have very similar spotting patterns, and so it probably has to do with it's just a common it's more common to have that overexpression of the melanin in those species than in others. Yeah, I mean you could get a melanistic anything. We have melanistic house cats, right? So you can make it happen, but it probably also has to do with the environments that they can just survive well in the tropical rainforest environments, and they don't in other places. So you may actually you probably do get melanistic wildcats in the southwestern U.S., but they're surrounded by tan. It's like they're not going to survive well. They're going to get picked up by a hawk or something really early on, which would be awesome to see. But I'm sure you have that because there are melanistic gray tree squirrels, and there are melanistic. I mean, there's melanistic forms of almost every mammal you can think of. Yeah.
Wait, so is someone being melanistic considered a mutation, or is it just a variation?
Well, both. It's a variation that is a result of a mutation in the amount and how the melanin gets deposited.
Because like I know albinism is definitely a mutation, but like,
Yes.
How do you know the difference between whether something is just a variation that's in the genome, or if it was a mistake, kind of?
Well, you probably answer that there is no variation apart from mutations at one point or another, right? Unless you believe that a variation was designed or it's some kind of an epistatic effect. But that you would have to be able to show that it's some kind of an epistatic effect and not a change in the genome itself. Yeah. And so here's is where you don't get the apoptosis, and so you have an issue in the apopstatic pathway, where in development the cells in development your fingers start to develop in pads that are all just together. And then you have programmed cell death happening in between them to actually separate the fingers. And so here you have the results of a mutation in an apopstatic pathway in which you don't actually get that separation. And you could also look at the number of digits you have here, because this is more than just a single digit, that this is also polydactyly, which is another mutation. Yeah?
Is that like the same thing that happens when people only have like one finger?
Not necessarily. Yeah, not necessarily. There are a lot of things that can developmentally happen. And so here's sickle cell trait or sickle cell disease. It tends to be declared as the best example of a beneficial mutation. And that is that it's only beneficial in heterozygous individuals, and that it gives them some protection from developing malaria. Yeah, Sadie.
I had a question relating to the hand.
Yeah.
Are there still, do they still have all of
Yes, usually. Yep, and usually you can correct it pretty easily, right? You just go in and you surgically separate the digits.
Yeah, and the same thing happens with legs too, right? Where
Sure.
there's
Yeah, I
that
mean anywhere.
separation between the limbs.
Sure. There it's a little bit different because the both hind limbs don't develop as a single piece like the hands and the feet do. They develop as separate buds that then develop into the lower limbs. But so for there, you'd have a different mechanism by which they were attached rather than failure to complete the apopstatic pathway. And then the final example of mutations, right? I did this last year and it went over really poorly, so I decided to keep it in here. Oh, you gotta love it. You gotta love it. All right. How is DNA packaged within the cell? And remember, I told you from the quiz, there are three main ways that proteins interact with the DNA. To copy the DNA, to express the DNA, and to organize the DNA. And so how is DNA packaged within the cell? The easiest way to answer that question, you would say with the help of proteins. How is DNA packaged within the cell? With the help of proteins. And so, just as the replication of DNA involves a number of different proteins interacting with the DNA, the organization of DNA requires several different proteins interacting with the DNA molecules. And so we have DNA interacting very closely with proteins in eukaryotic genomes, forming what we call chromatin. And so chromatin is a molecule that is a combination of nucleic acid and protein. And so that is the norm for eukaryotic chromosomes, to have proteins intimately involved in organizing these chromosomes. And so some of those proteins, histones, and these are made up, these are basically octamers. So you have 4 different polypeptides, H2A, H2B, H3, and H4, 4 different polypeptides, 2 copies of each combined together into one protein with quaternary structure. So we call it an octamer, a single functional protein with 8 polypeptides. And these form the histones. And so the DNA actually forms ionic bonds around these histones. Histones are positively charged, the DNA is negatively charged, and the DNA wraps around these histones, these protein octamers. And so as it does that, it condenses the DNA down. Have you ever heard that if you took the DNA out of an average human cell and you just lined it out, it'd be 6 feet of DNA in every human cell? And you're like, how do you package that into a space the size of the nucleus? And that the answer to that is you wrap it tightly around small proteins. Yeah, Alyssa.
How does the DNA not get ruined when it's bonded by something else? Is the bond weaker than the hydrogen bonds?
That's a good question. The bond isn't weaker. Ionic bonds are stronger than hydrogen bonds individually. But the ionic bonds aren't strong enough to overcome the full weight of all the hydrogen bonds. No. Yeah.
Do you ever have it where the histones are like so tightly bonded that when they try to read it that it breaks?
Oh, so yeah, when DNA is wrapped around these histones, and these nucleosomes, these are the complex of histone plus DNA. Histone plus DNA together is the nucleosome. When the DNA is wrapped around that, you can't do anything with that DNA. You can't replicate it, you can't express it. So if you want to replicate the DNA or you want to express the DNA, you have to unwind it from that histone. You actually have to physically take it off the histone. And so there are proteins associated with doing that. So there are proteins keeping it organized, wrapped around the histones, and then there are proteins that will unwrap it so that you can actually work with it. And that's actually this last question. So how can DNA be replicated when it's wound so tightly around these histones? And so the answer to that is you basically have to unwrap it. And some sections of the DNA are just naturally loose, more loosely connected than others. More loosely connected than others. Man, I gave you all this great hope. And I'm like, we're going to finish chapter 14 today. And, we're going to, whenever we finish, we'll be done, even if it's before class time. And we're not going to finish 14 today. And we're out of class time. Well, it's still 12, now it's 12:40. And so we're out of class time and we didn't finish chapter 14. We've got like maybe 2 minutes worth of it on Monday to deal with. All right? So on Monday, I'll put the slides for chapter 15. I actually put them on today. We'll start going through that on Monday, chapter 15, which is all about expressing the DNA. So we've talked about copying the DNA, talked a little bit about organizing the DNA. We'll finish that on Monday, and then next week we'll talk about expressing the DNA. All right, have a wonderful weekend. Be safe. If you're going on Disney Day, have fun. If you're not going on Disney Day, still have fun.